Okay, so the first question is to list and describe 3 differences between gram-positive and gram-negative. So if we look at the cell wall pictures that I did, you can see that there's teichoic and lipoteichoic acid in gram-positive, none of that in gram-negative. Thick cell wall in gram-positive, thin cell wall in gram-negative. In gram-negative, you have an outer membrane with lipopolysaccharides, lipoproteins, and porin proteins, and you do not have that in gram-positive cell wall. You have a small periplasmic space in gram-positives. You have a large periplasmic space in gram-negative. So those are some differences. How the outer membrane of gram-negative bacteria contributes to disease symptoms. Well, the fact that it is hydrophobic prevents a lot of things from getting through, and even harmful chemicals, the porin proteins can change size so that the chemicals cannot get through, and so it's basically impermeable to some antibiotics as well, and the antibiotics that have to get into the cell wall or have to get past the cell membrane even to get to the proteins and to do damage inside the cell internally, they're going to be naturally resistant, especially if they're not able to get past that outer membrane. So that outer membrane protects the gram-negative bacteria. 3 functions of the cell membrane or cytoplasmic membrane. They are involved in energy reactions, synthesis reactions. They transport nutrients into and waste products out of the cell. So hopefully you had that discussion or you thought to yourself those answers. And now section 4. So section 4.4 is the bacterial internal structures, and we're going to look at 5 structures that may be contained in bacterial cytoplasm, and then describe or detail the causes and mechanisms of sporulation and germination and the process of germination and sporulation. So bacterial internal structures, they have all of them, all cells have a cytoplasm. It's mostly water. It's going to have sugars, amino acids, and salts in there. It's where all the biochemical reactions occur and synthetic activities. It also is the place where we have the nucleic acid, the chromosomes, the chromatin body, the nucleoid region, the ribosomes, the granules, the fibers that act as a cytoskeleton. So the chromosome is different. DNA in the bacteria cell is a single circular strand or loop of DNA. So it's in eukaryotic cells, it's multiple, and it's linear, and in bacterial chromosome, it's just a single loop of DNA. It's a very large loop, but for the size of the cell, it has to compact itself. So it does tightly coil around basic protein molecules to fit into the cell compartment. And so that is the next thing I'm going to show you, an animation on how that works. So here's the circular chromosomal DNA and how it compacts.
The circular chromosomal DNA of bacteria must be compacted about 1,000-fold to fit within a bacterial cell. Part of this compaction comes through the formation of loop domains, where segments of DNA are folded into loop-like structures that are held in place by DNA-binding proteins. The formation of loop domains compacts the DNA about 10-fold. Further compaction is accomplished through a process known as supercoiling. In this process, which is under the control of the enzymes DNA gyrase and topoisomerase I, twists are introduced in the DNA molecule that cause it to compact by coiling onto itself. This is much like the process of coiling that you can observe by twisting a rubber band. DNA looping and supercoiling make the bacterial chromosome more compact so that it can fit within the nucleoid of the bacterial cell.
So plasmids are non-essential pieces of DNA. They're kind of like what I would call extra credit. You know, they help them survive, just like extra credit helps you survive this class. It's separate from the chromosome. They're double-stranded, but tiny circles, smaller, definitely much smaller than the chromosome. They're duplicated and passed on to offspring during replication, and they often will have confer protective traits. So some of the traits, they may produce a chromosome, or they may produce a capsule. They may be involved in antibiotic resistance. They have many different functions. They could produce a toxin, and so all of these things may help them in some way to survive. And so they're an important agent in genetic engineering as well. So we can insert genes into a plasmid and then insert the plasmid into a cell, and it can then start to make things for us. So this is basically how insulin is made. They have recombinant plasmids where they took a piece of a gene from human cells and put it into a plasmid and then inserted it into E. coli bacteria. And now E. coli is a cell factory for insulin. So ribosomes are made of RNA and protein. They're in the cytoplasm. They're also located they're located in the cytoplasm. There's no rough endoplasmic reticulum in bacteria cells. And they're measured by Svedberg units, that's the S, and that is a measurement of the relative size of the cell parts through sedimentation during centrifugation. So it has to do with kind of the size and the density of the particles as to where they're going to sediment during centrifuging. And so the bacterial ribosomes are 70S when they're together. When the two subunits are together, the large subunit is a 50S subunit and the small is a 30S subunit. And when they come together, they will centrifuge at a 70S sedimentation. Where the eukaryotic cells have ribosomes of 80S when they're together, and the large subunit is 60 and the small unit is 40. So also inside the cytoplasm we have inclusion bodies, and these are for storage during periods of abundance. They'll store up this particular drawing, it's not a drawing, it's an electron micrograph that's been colored. The MPs are magnetic particles, and they use this to navigate through and along lines of electromagnetic radiation, so they can move along the magnetic poles on the Earth. And so they vary, these inclusion bodies vary in size, number, and what's inside them. And the cytoskeleton, you can see here, it looks like in little helices. This almost looks like spirogyra, but they're arranged in helical ribbons around just inside the cytoplasmic membrane, and they do contribute to the cell shape. They're a potential target for antibiotic development, because if we can destroy the cytoskeleton, then we could possibly make them, destroy the cell. So this could be a new target, potential new target for antibiotics. But they are the same kind of like proteins, similar to what we see in eukaryotic actin. So the bacterial endospores, which we didn't see a picture of in the diagram earlier in the chapter, but an endospore is for survival. It's not a reproductive structure like we see in fungi. It's not an asexual or sexual, either one. It's not for reproduction. Reproduction would mean that one cell produces two cells. But this spore, the cell forms one spore within the cell, and that spore is going to withstand the hostile conditions, and then when the conditions are better again, it germinates into one cell. So one cell makes one spore will make one cell. You didn't increase in number, so it's not a reproductive thing. It's a survival mechanism. So the vegetative cell is the metabolically active part, and the endospore is the resting condition, the dormant, inert part. And sporulation is that process of making a spore, and it's induced by environmental conditions. So when the conditions are bad, whatever bad is, so for an aerobic bacteria that needs oxygen, when it's in an anaerobic environment, that's bad, and so it will maybe sporulate at that point. When you have an anaerobic bacteria, where if it's in an aerobic environment, then that's bad, and so it'll start producing spores in an aerobic environment. So they can resist a lot of conditions like heating, drying, freezing, radiation, chemicals. They just, the radiation, chemicals can't get through the spore coat, and heating and drying are not going to affect them because of that spore coat. So the stimulus is a lot of times depletion of nutrients, especially lack of carbon and nitrogen. And so it takes about 6 to 8 hours for most species to form a spore. And this is the basic process, but I'm going to give you a 6-step process that'll be a little bit, or 5-step process that'll be a little bit easier than 9 steps. But you can see that, the chromosome is duplicated. So here's the normal cell, vegetative cell, and the chromosome is duplicated, and it invaginates and forms like 2 compartments inside the cytoplasm. So the cytoplasm is divided asymmetrically into a forespore, which is smaller, and a sporangium. The sporangium, or mother spore, engulfs the spore. So the larger section engulfs the smaller and forms that, what would be called the cortex. So the cortex is, technically it's a cortex when peptidoglycan is laid down, and then the cortex is coated with some protein layers that are deposited outside of the cortex. So peptidoglycan is laid down in between, so you start with 2 layers because of the larger one engulfing the smaller, and then peptidoglycan is laid down in between those layers. That would make the cortex. Then the spore coat forms around the cortex. And at that point, it's called a mature endospore, and the vegetative cell starts to degrade and dies off, and you can see the DNA kind of degrading here. And then the vegetative portion is releases the spore, or it just degrades. And then eventually, when the conditions are good again, the cell will swell and it releases the endospore, or releases the vegetative cell.